Okay, so hello, good evening, everyone. Welcome to today's session. Today we will be discussing, we'll be continuing with our discussion on genes, genomes, and the epigenome. And as you remember, this is week 8 of our classes, and we have begun a discussion on epigenetics and the basic concepts of epigenetics in the previous session. So previously we had followed the given outline for the course where we started off with our discussion on gene structure, function, and regulation, mutations, polymorphisms, the difference between the two, and how functional genomics has a role to play in the same. Then we moved on to our discussion on using Sanger sequencing, on next generation sequencing, and the difference between these two techniques. And then we moved on to the topic of unraveling the genome, exome, and transcriptome, where we discussed in depth about the pipeline used for whole exome sequencing, and we briefly understood the differences between whole genome, whole exome, and transcriptome sequencing, and how you can use different strategies based on the question in your hypothesis that you're trying to address. So from there, we took a little bit of a detour because we had a lot of classes which discussed about these sequencing methods and their analytical tools, etc. And then we moved on to this one, which is for Introduction to Epigenetics and the key concepts associated with the same. Today, we'll be discussing histone modifications in a lot more detail. We'll be continuing with the topic we had already introduced in the previous class, and we'll go into more detail about histone modifications. And in the next class, we'll move on to DNA methylation and the strategies which are used to map DNA methylation in the genome. Some introduction you'll already get in this class, but details will be taken up later on. And from there, we will move forward to analysis using these tools so that from there we can move on to the techniques used for epigenetic and epigenomic profiling as well. So, with this plan, I wanted to quickly revise what we have already discussed in the previous class so that we are, updated on the topics which were discussed then. So we originally started off with introduction to epigenetics and some of the key concepts which are related to the same. And if you remember, I had already referred this book on chromatin and gene regulation by Brian M. Turner. This is the book I had referred to in the previous class. It's a great book for beginners, and especially if you're somebody who is just getting familiar with epigenetics and basic concepts, and you have some background in molecular biology. It's a wonderful book to actually start off with. You can refer to any material available online, anything which is suited to your understanding. In today's class, I have taken some very few elements from this book, but mostly what I'll be discussing today has been taken from review papers and journals, which I'll give reference to later on. And you can use any of that material. You can use any resources via PubMed, any books in your library, whatever is comfortable for you. So, with this, let us quickly recap what we discussed in the previous class. So we started off with the fact that there's a very close interplay of gene regulation and epigenetics, even though it seems like epigenetics is a epigenetic modifications are sort of a superficial mark which is left behind. That's not actually the case. And in fact, epigenetics has a very crucial role to play in controlling gene expression, in controlling gene regulation, and in fine tuning it based on the context in which the cell is trying to address gene expression at that point of time. So, well, the main concern that the cell is actually addressing is the fact that it is able to switch some genes on and off as per the requirement and in response to some signal which has come from outside of the cell, which is necessary because it should be responsive and sensitive to external signals so that its own function can then be controlled. Um, in addition, you also have to keep in mind that if gene expression was not or was only controlled by trans factors, that would also leave a lot of vulnerability open, and also it would not always be feasible to maintain that particular control over generations because it would be dependent on the binding of certain factors. But instead, an epigenetic mark left behind is a covalent change that has occurred in the either the DNA or the histone which is attached to the DNA, and therefore it is something which is heritable over generation and heritable from the parent individual into the gamete, and therefore can be transmission can be um, taken forward over generations and can be inherited from the parent to the child. Now, one very important concept that is relevant for epigenetics is that chromatin packaging and chromatin um, formation of chromatin fibers from loose DNA is something which is extremely necessary considering the magnitude of information that needs to be contained within a very small cell. So every nucleated cell, it contains near about 2 meters of DNA, and that is an immense amount of information. If you think about it in terms of think of one cell as a computer system and think about how much information needs to be available to the cell and should be available in its random access memory or its RAM so that it can be used as and when the cell requires it. So all organisms, whether they're prokaryotic or eukaryotic, actually have to deal with this problem of packaging a lot of information or a lot of DNA into a very small space within the cell. And another very important factor is that that information there should be accessible, and therefore the information should be functional. It should not just be a static macromolecule lying around in the cell, but it should be something that can be expressed, that can be repressed, that can be controlled, and that is sensitive to external signals and is responsive to the environment. Secondly, the cell must be able to replicate with accuracy just once in each cell cycle. The copies of DNA should be segregated into daughter cells every time the cell undergoes division, and the genetic information that is encoded within the DNA should be expressed in a way which is appropriate to the condition that the cell is enduring at that point of time. So proper packaging of the chromatin therefore becomes a very challenging and b very crucial task that needs to be perfectly performed so that gene expression is not adversely harmed. Next, Next, how is this packaging actually done? Physically talking about it, it is actually a DNA and protein interaction which is able to facilitate this. So one example which was given in context of the experimental validation for the same was that um, if you're performing DNA isolation or if you're isolating the nucleus from cells or from any patient tissue, and then you're extracting all of the proteins, then almost half of the proteins extracted would actually be these DNA binding moieties, which are called as histones. So these histones would have been known for hundreds of years, and they were first described by Albrecht Kossel in 1884. They're very small proteins. They are 10 to 12 kilodaltons in weight, and they're very highly basic. By basic, we mean the type of amino acids which are the most dominant component of histones are lysine and arginine, and they are positively charged at physiological or at neutral pH. They're not present in prokaryotes, but there are some proteins which are histone-like proteins which are present or that have histone-like structural motifs. Most of the discussion, or rather all of the discussion will be carrying out here, unless otherwise mentioned, is in context of eukaryotic organisms. And when we talk about histones, we are majorly referring to these out of these five, we are mainly referring to four of the varieties, which are H2A, H2B, H3, and H4. H1, as we have previously discussed, has a different job to do. It sort of acts like a demarcator or like a stopper for the formation of a nucleosome. Now, each of the residues, the information or the properties about these histones have been mentioned here. You can see how many residues are present there, their molecular mass, and the percentage of the residues that is actually dominated or that is controlled by lysine and arginine, and what is the approximate positive charge. So you can see that all of these stones are positively charged, and a good percentage of them is actually predominantly lysine or arginine, and therefore the charge is, and that is how the charge is imparted onto the histone protein. Um, what we also understood in the previous class is that most of the experiments that you see for epigenetic understanding majorly involve the disruption of these DNA protein interactions, not at the interface, but at the exposed areas or at the so-called euchromatin, where DNA is not protected by the bound protein, and such regions can be digested or can be cut or can be digested by certain enzymes like micrococcal nuclease or MNAs under certain conditions that allows MNAs to enter into the nucleus. Now, most nuclear DNA is susceptible, that is approximately 85% of nuclear DNA is susceptible to this pattern of cleavage, showing that the repeated packaging unit is actually this nucleosome. Now, what exactly do we mean by this nucleosome? So it most primitively or most early on, the studies which observe that DNA under the microscope looks somewhat like this beads on a string sort of a structure, like a pearl necklace, and there are many different names to it. Actually, if you read initial descriptions made by scientists who made these very unique observations, mostly they tried to relate it to something which could be understood and which could be interpreted by people, by the common public. Like, for example, um, some initial understanding of the cell, for example, was thought to be like that of a watermelon or like that of a pie, where all the electrons and all the electrons are sort of scattered throughout the watermelon, and therefore it doesn't have any random organization, etc. Then it under more and more understanding made it a lot more aware, a lot more accurate, and of course, imaging is something that has significantly improved understanding in the past few decades. So, with this in mind, this beats on a string structure is something which has stayed relevant even today, because it does actually have a lot of, it actually does tally to a large extent with the realistic scenario where it resembles this beads on a string sort of a structure. So, yeah. So, this, if you remember, is the structure of DNA, the structure of the double helix, whereby you can see that there are two dominant architectural motifs. One is the minor groove, one is the major groove, which are sites for binding of transcription factors, which allows them to grip or use the DNA as a scaffold and bind with, good, with high precision, with high accuracy. And approximately one complete turn is said to have 10.5 base pairs. It's mathematical calculation which gives that 0.5, because there's one base which is partially a part of one turn and partially a part of the next. So that is what the dimensions of the DNA double helix actually look like. Now, uh, chromatin fragments can be separated into units of discrete sizes by the process of centrifugation. So if a chromatin preparation is layered on top of a sucrose solution in a centrifuge tube and then spun at 10,000 G, chromatin fragments move down the tube at different speeds. And what you effectively end up getting are these fragments where you'll be able to understand what the pattern of DNA that is bound actually looks like. And also what you see that the chromatin bands can be separated into small fractions, and when their optical density is measured, you'll be able to see what it looks like in terms of the peaks. And in C, you can observe that if the DNA from each of these peak fractions is then resolved by electrophoresis, you'll be able to see the DNA that is bound there. And then finally, in point D, you can see the protein electrophoresis for all the four histones, and you can see that there are four core histones that are present in equimolar amounts, and that is what it looks like. So, with this information, we went ahead and understood the structure of the nucleosome. If you digest the nuclear material with micrococcal nuclease under mild conditions, and then you separate, you'll be able to see that histone H2, H3, H4, and H2, H3, sorry, H2A, H2B, H3, and H4, they separate out, and there is one separate histone, which is histone H1, which is exposed to, which is dissociated from chromatin by exposure to salt concentrations around zero point 0.4 molar. And yeah, that's what we observed in the bands also. Now, when we come to define the basic fundamental or repetitive unit of chromatin, it basically involves 200 base pairs of DNA and um, a core histone octamer, which is made up of two of these four core histones, H2A, H3B, H3, H2A, H2B, H3, and H4. And the histones form this octameric structure, which, along with near about 175 to 200 base pairs of DNA, is able to constitute a nucleosome. Now, a chromatin fiber, when we talk about that, actually consists of many such nucleosomes, which form a flexibly jointed chain. This corresponds to the bead on the string structure, like we previously mentioned. The histone octamer can actually be dissociated, and what you will get are these dimers of H3, H4, and H2, H2B. And the first experimental clues came from nucleus digestion studies, where people saw how nucleosomal DNA was digested by DNase 1, suggesting that DNA is actually coiled around the octamer. Why this is relevant is because depending on how the DNA the nucleosomal DNA and the histone actually interact with each other. Based on that, the exposure to different types of nucleases, such as MNase, can actually be decided. Now, while DNA MNase is able to make double-stranded cuts in DNA, it needs to actually straddle the DNA, it needs to sit on the DNA and then make the cut. Sorry. Therefore, the enzyme is not able to cut DNA, which is resting on a surface, which explains why nucleosomal DNA is protected from MNase digestion, even if it is located on the outside of the octamer. In contrast, when we try to understand how DNase 1 works, you can see that it preferentially cuts DNA at sites which are farthest away from the histone core. And there is a preferential single strand cutting, which is also done, which is indicated by arrows. And then you can resolve it on a gel, and you'll see that DNase 1 treatment differs in size by around or approximately 10 base pairs, suggesting that there's a periodicity in this digestion as well. And the way in which it does is that it can make single-stranded sorry, single-stranded nicks, but only the DNA which is farthest from the surface can be easily cut under these circumstances. Just excuse me a moment. Okay, yeah. So, DNase 1 operates differently. Therefore, you see a difference in the pattern of digestion by DNase 1 and MNase as well. Also, what you see here is the linker histone. Like I previously mentioned, it occupies a position right outside of this core octamer, close to the point where DNA actually enters and exits the nucleosome, which is consistent with the effects you see on nucleus digestion as well, because nucleus is able to access it also. And this nucleosome complexed with H1 together is sometimes referred to as the chromatosome. So nucleosome plus H1 is chromatosome. Histone octamer plus the bound nucleosomal DNA together makes the nucleosome. So that nucleosome along with linker histone H1 together makes the chromatosome. So that is the flow chart. What you see here is the histone handshake, which, as we previously discussed, is one of the most sort of predominant motifs which is observed in the histone octamer. Now, before we get into details of this, a quick recap that there are four types of these core histones like we have discussed: H2A, H3B, H2A, H2B, H3, and H4. And if you look closely, you'll be able to see one repetitive motif, which is also often called as the histone fold, which is the presence of three alpha helices in tandem with each other. They are usually denoted as alpha 1, alpha 2, and alpha 3, the three helices. And along with that, you can see there are some motifs which may or may not be different, and they are majorly present in H3 and H2B. Also, one more thing to note here is that two of these are slightly longer histone proteins or polypeptides, which are H2A and H3, whereas H4 and H2B are relatively shorter. The central globular domains of all the four core histones at this alpha helix, like I already mentioned, as you can see mentioned here, the broken lines represent alpha helical regions which are not a part of the histone fold, which is shown here in light gray. These fold domains, the histone fold domains of H3 and H4, interact in the core particle, and they then form this unique structure which is called as a histone handshake. It's much like if you bind your if you put your left and right hands together, the sort of orientation that they would follow is what is actually the composition of the histone handshake as well. The histone the histone octamer has three helical regions in each of these four core histones that are arranged in this very similar manner. Specifically speaking, H3 and H4, they interlock, and H2A and H2B, they interlock to form this histone handshake. That is one of the reasons why you'll get these dimers of these individual motifs to form the complete octamer eventually. It is this handshake interaction that gives rise to the stable dimers that we have already mentioned. Now, this histone tail part is something I'll discuss in more detail in today's class, so I'm skipping it for now. Apart from that, we also started off our discussion on modifications. Again, this is something we'll discuss today in more detail. But majorly, the most common types of modifications that occur uh, are on the histone tails because they have really long N-terminal tails, which are not a part of the octamer, and they sort of protrude out through the DNA or between the DNA, and therefore they have a major role in controlling how DNA expression can proceed. Commonly speaking, this includes modifications like acetylation, phosphorylation, methylation, ADP ribosylation, sometimes also ubiquitination and sumoylation. Today, I'll also introduce another type of modification, which we'll come to later. It's called crotonylation. As you can understand, acetylation means the addition of an acetyl group, and deacetylation is the removal of the same. As you can see here, there are some enzymes which are actually able to do this job, and therefore their expression is what controls epigenetic modification as well. So when you are carrying out epigenetic profiling, this is what you will try to understand. You'll try to see the global change in acetylation or methylation. But yeah, most commonly what is studied is DNA methylation, like you can see here. In contrast, if you're studying histone modification or histone methylation, then that is going to that has to be approached by some different methods. We'll again come to that detail later on. Basically, the idea of acetylation and deacetylation is the fact that acetyl group imparts a negative charge onto the histone, which is in contrast to the high net positive charge which histones carry by default due to the abundance of lysine and arginine residues. And therefore, as a consequence of this, what you expect is that the interaction, or what is the implication is that the interaction of the DNA molecule and the bound histone is somewhat well, not compromised, but is somewhat reduced due to the addition of this negative charge, and as a result, the less strong or the weaker interaction is able to control the reduce or the repression of transcription and the repression of gene expression and the activation of gene expression, because DNA is now less strongly bound and more of the DNA is now exposed. More of the DNA is now euchromatin instead of heterochromatin, and therefore transcriptional activation can take place. Now, when this addition or removal of acetyl groups occurs at some specific genomic loci, like for example, in the control regions of the in the expression control regions of certain genes, then that can actually have a very important role to play in overall controlling the expression of that gene. Now, keep in mind the control does not necessarily have to be immediately upstream. This is exactly where our initial classes actually have a role to play, where we understood the concept of insulators, enhancers which are in close proximity or are close to the transcription start site, some enhancers which are high upstream or high downstream. All of these different subtypes are possible, and it depends upon where the or how the 3D structure of the chromatin is actually formed, because that is something that has a major role to play in final expression, and it is something we'll discuss later on as well. But that dominantly controls gene expression also. Additionally, a second type of modification, which also has a major role to play, is methylation. And most commonly, you'll find methylation to be um, controlling H3 and H4. And methyl lysine is also very chemically stable. And modification is also complicated by the fact that multiple methyl groups can be added. The donor in this case is S-adenosyl methionine. We've already discussed the difference between DNA and histone methylation. So I'll move past that so that we can begin today's class. I think this was our last topic for yeah, the previous class, where we introduced histone variants. What this basically means is that histone genes, um, commonly, while we understand those 5 different histones to be present within a cluster, they are not the only possible option, and instead, you have certain alternatives or certain special histones that are expressed temporarily or that are expressed in response to certain changes, in response to certain environmental signals, in response to certain physiological processes, which is enhanced or is supported by the expression of this histone variant. And in some if you compare the sequences or the domain organization or the structure of these histones, you will see that in some cases these differences are not very intense. There are some key amino acid substitutions, like you can see here a comparison between H2A and H2AZ and macro H2A. So if you look at H2AZ, it only has some base substitutions. That does not really completely transform the nature or the function or the properties of that histone. Like for H3, for example, you have H3.1, 3.2, 3.3. The difference is usually in context of a single amino acid. Like for example, serine to cysteine at position number 96, whereas H3.3 has 2 additional substitutions, whereas H3.1 and H3.2 can only be synthesized during the S phase. In contrast, you'll find some cases where there are major domain alterations. For example, H2AZ differs from the human variant H2A, H2A.1, which is by 15 amino acid substitutions. Now, that is a lot, and they are sometimes also supported or sometimes also demarcated by insertions, deletions, substitutions, etc. A second histone variant, H2O variant, which is histone H2A, here, like you can see, is quite different. It also has over over 200 additional amino acids, which differs very largely, macroH2A. And it also has been found to associate specifically with inactive or transcriptionally silent or heterochromatinized chromatin. And therefore, macroH2A is often involved in heterochromatinization. So therefore, when such, because it possibly has certain substitutions which um, in, immensely alters the interaction of DNA and histone, and therefore makes the interaction very strong, and therefore significantly represses gene expression in those loci. So these are just some examples. Please do not consider any of these cases to be very rigid. It depends upon what is the need of the cell, what is the most um, accessible sort of variant, and what is the way in which that particular variant actually works. Coming to the next class, which is today's session, we will understand histone modifications in more detail, and we'll take one example of histone methylation mark, which we'll understand in more detail today. Okay, so the initial few lines are just a recap of what we've already discussed. The fundamental unit of chromatin is the nucleosome, which consists of the central histone octamer. Yeah, the histone octamer, which has to each of the histones H2A, H2B, H3, and H4, around which you'll find nearly 1.75 left-handed superhelical turns of DNA. The histones are decorated by a plethora of post-translational modifications or PTMs, often referred to as epigenetic marks. This is a very important terminology. You'll find it very commonly used in literature, in papers, anything you refer to. Just a second, my switched. Yeah. So it is something which you'll find very commonly referred to in papers which are related to epigenetics and paper which are related to histone modifications or DNA methylation, etc. So epigenetic marks are something which have been left behind by epigenetic modifiers, and that affects the eventually affects gene expression and gene regulation in the long run. Um, Histone PTMs or post-translational modifications have been often suggested to serve an epigenetic code. What do we mean by that? It means that there is a difference between the genetic code and the epigenetic code. By the genetic code, we mean the sequence within the DNA which has undergone transcription into an RNA can actually be designated into or can actually be decoded in the form of proteins based on what is the sequence of nucleotides which are present in the mRNA sequence, and therefore each triplet of the nucleotide sequence corresponds to a particular type of amino acid. That is what we understand by the genetic code. And if you remember in our previous classes, we had discussed different properties of the genetic code, like the fact that it is continuous, it is non-overlapping. The codons, they are degenerate in nature. They're mostly universal. Some exceptions are some mitochondrial proteins and certain prokaryotes, etc. So these were some of the properties we had discussed. In contrast, when we talk about the epigenetic code, as the name obviously suggests, it's not the DNA sequence, but it is the changes or the information that is decoded above the genetic sequence, which is imparted by post-translational modifications which are occurring within that DNA sequence. So the diversified, histone PTMs have even been suggested to serve an epigenetic code, in which individual marks the type of change that has occurred within the DNA. Each of those marks have their own message to convey, and also the diversified modification of histone amino terminals or the N-terminal end expands the genetic code, so it is called as the histone code. So because this epigenetic mark is very predominantly contributed to by the histone N-terminal tails. Therefore, it is often also, instead of being called as the epigenetic code, sometimes it's also referred to as the histone code. So these are the four most common types of modifications that are acetylation, methylation, phosphorylation, and ubiquitination. You can see that H2A, H2B, H3, and H4 for each of them. The position has been mentioned at which the most common type of modifications occur. And as you can obviously understand that the nomenclature or the order in which this has been referred is that protein chains are normally, or the amino acid or yeah, the amino acid part forming the polypeptide is usually labeled from its N-terminal end, and because the N-terminal end projects out, the base 1, 3, 5, etc. is projecting out, and base 120 is projecting in or is a part of the octamer, possibly a part of the octamer. And you can see that there are different, completely scattered sort of modifications that can occur in each of these loci, whether it is acetylation, methylation, phosphorylation, ubiquitination, and so on and so forth. So this actually is what forms the histone code, or what forms the domain of epigenetic modifications that can control gene expression and gene regulation. What you should also just give me. What you should also look at here is that there is a lot more detail about the types of tails which are present, whether it is H2A, H2B, H3, and H4. You can see some representative images. Each of these marks denotes something else. You can find the detail of that here. So I'll just quickly mention what are the different types of marks which are present. Some of them are acylations. They have been, further subcategorized. Lysine, PTMs, and other amino acids as indicated. The other amino acids or alternative amino acids have been indicated within the bracket. K is for lysine. If you're familiar with the one-letter code for amino acids, K is for lysine. S is for serine, T is for threonine. Mostly you'll find these. R is for arginine, and so on and so forth. So the different types of modifications are largely classified into either A, acylations. Acylation includes formyl group addition, acetyl group addition, propynyl group addition, butyrylation, crotonylation, something I already mentioned. It's something you might not find traditionally in older books, but recent research papers in the past couple of years, they have discussed this. Research and review papers. Then you have benzoylation, you have 2-hydroxy isobutyrylation, hydroxybutyrylation, lactylation, malonylation, succinylation, glutyrylation. What this basically means is that a certain side chain, a certain substituent group is added onto one particular type of histone, and it has some effect on gene expression and gene regulation. You also have ubiquitin-like groups like ubiquitination, somylation, alpha ufmylation. Others include methylation, biotinylation, ADP ribosylation, etc. Non-lysine PTMs include serotonylation, dopaminylation, O-palmitoylation, S-palmitoylation, isomerization, hydroxylation, glucnac group is added. Then you have deamination. So that is what you see here. You also have phosphorylation, which can occur for four different types of amino acids: serine, threonine, tyrosine, all of these different groups, and also histidine, and also N-terminal acylation, which is only seen in case of serine. So these are the different types of groups. Now, within each amino acid sequence, you can see that there are certain patches which have been indicated. These are part of different histone or different histone entities or histone bodies, and you can see that there are certain residues which carry a huge range of modifications. So this actually enhances the rate of control which is executed by these epigenetic modifiers, which are able to add or remove a certain type of group. Also keep in mind, it's not necessary that they have to occur in an order, that certain modifications can be added after another or before another, or cannot be added in at the same time, or they are mutually exclusive, whatever are the different types of alternatives that you can imagine. They are all going to be very context specific. So certain bases, in particular here you'll find lysines have been indicated. They carry around 10 to 12 different possibilities of modifications, and sometimes these modifications are not sometimes these modifications are actually read as epigenetic marks, and sometimes it is the presence or the absence of a sole epigenetic mark or of a combination of epigenetic marks that can largely alter the way in which gene expression can be carried out. So again, please keep in mind that different types or different frequency and the effect it manifests can sometimes crosstalk with each other to make the to amplify the signal a lot further. Oh, okay, keep it off. Next, what you need to understand in this continued concept is that the histone PTM landscape, or the post-translational modification landscape, is laid down, maintained, and reset. What do we mean by that? By laid down, we mean the fact that these modifications are necessary and they are controlled or they're regulated by certain types of signaling or certain types of pathways, and it is something which is the default within the cell. Second, their sustained presence or their maintenance is another very important context. Third, um, yeah, Third, numerous interconnected signaling pathways can actually operate in tandem, and that involves crosstalk between multiple signaling. For example, okay, we'll come to the example later. Multiple signaling pathways can dictate whether a certain gene needs to be expressed, whether a certain gene needs to be hyperactivated or repressed, or if we talk about a disease concept. So for example, again, using the same example we usually refer to, that is in context of cancer. Sometimes epigenetic modifiers have an immense role to play. Sometimes they show some oncogenic activity, or they show immense roles in acquisition of drug resistance. So all of these are or their expression, um, or their incorrect functionality is often the consequence of hyperactivation or the downstream manifestation of multiple signaling pathways that have gone wrong. Again, a very context specific thing. Next, the fact that these sort of modifications or this control of epigenetic changes is very largely modulated by a group of enzymes that can be further like they can be commonly referred to as epigenetic modifiers, but they can be further segregated into their subtypes based on what activity they actually carry out. Like some might be writers, which are adding a certain modification. Some of them can actually be readers, which are able to identify the type of epigenetic mark that has been left behind, and some of them can remove PTMs, which are erasers. Histone PTMs are present in both the terminal tails of histones and their globular core domains, and these different types of PTMs can exert their effects via a multitude of mechanisms. Keep in mind here we have only talked about histone PTMs that are present in the terminal tails because they have a major role to play. Also, because the 10 terminal tails are very long and they are protruding through the histone, through the nucleosomal core, Therefore, they are a lot more accessible to the epigenetic writers or the modifiers, which are able to add the epigenetic mark or add the epigenetic change. Uh, an inaccessible area is a lot more difficult to modify and is therefore not as susceptible as something which is present on the tape. Histone acetylation levels, for example, are co-regulated by two types of enzymes. One are, of course, the histone acetylases or the HATs, and the histone deacetylase or the HDACs. What are they in general terms? They can be classified or they can be also understood as HDACs, can be understood as erasers, because they are removing an acetyl mark or they're removing the epigenetic mark like we have previously discussed. And HATs are histone acetylases, so they add an epigenetic mark, so they are writers. HATs are relatively distinct from each other, and HDACs can be also classified into class 1, class 2A, class 3B, 2B, 3, and 4. The identification of mammalian HATs and HDACs, bromodomain containing domain. Okay, I think that's some error. But the identification of mammalian HATs, which are writers, HDACs, which are erasers, bromodomain containing proteins, not domains, but proteins, which are known as readers, and HDACs have laid the foundation for in-depth studies of histone acetylation modifications. For example, if you look at um, this sort of organization, think of it like a subsection of a chromatin fiber, which is very tightly packaged. As you can see here, the histone octamer is located in, or different histone octamers are located very close to each other, and they're forming this sort of continuous chromatin fiber, which of course has the octamer at the center, and it is bound by the DNA towards the outer ends. And this includes, so they're present in this manner and they can actually be acetylated by HATs, which are also called as histone acetyl transferases, and they include different types of enzymes like those from the Gantt family, CBPP300 family, MIST family, and other acetyl transferases as well, or acetylases as well. In contrast, if you look at the deacetylase class, that includes class 1, class 2A, class 2B, class 3, class 4, and other enzymes. So if you look at the schematic a little closely, you will see that the chromatin in the upper panel figure or upper figure panel is a lot more closely packaged, a lot more dense, whereas that which has undergone acetylation is sort of opened up and is a lot more loosely packaged and has a lot of acetyl groups added to it. So this is the result of the addition of the acetyl group, which opens up the DNA, makes it a lot more loose, and as a result, a lot more accessible to RNA polymerase 2, which is able to activate transcription. So yeah, that is pretty much what happens there. Next, if we look at histone modifications and the effect they have on physiological processes, they have a role. It's not just transcription, but there are also other sort of modifications or other sort of processes which they can influence. For example, if you look at this sort of pie chart or this graphical abstract, you'll see that the first example is of transcriptional activation. Sorry. Where some common modifications that influence this process include acetylation and methylation at certain specific loci. Then you can see that for DNA damage repair, okay, yeah, here you can see this is a good representation of the histone octamer. But in DNA damage repair, we have H2AX, like we previously mentioned, the histone variant having a role to play. Its phosphorylation of its serine 139 is another sort of modification. H3K4Me2, H3K36Me3. Cell differentiation includes H3K4Me3, H3K27Me3, transcriptional inhibition by different methylations. Tri and dimethylations are different histone lysine residues. Cell mitosis by phosphorylation. Please keep in mind that certain types of modifications, they actually predominate certain physiological processes. It's not that they are exclusive, but they predominate the process. Like, for example, signaling within the cell, any survival associated signaling, and cell cycle, cell division, these processes, they are predominantly under the control of phosphorylation dependent or phosphorylation associated modifications. Whereas if you talk about gene expression, majorly you'll find the dependence to be on acetylation and methylation, or rather on acetylation. In when we talk about gene silencing, it is majorly predominated by um, methylation. So it is the relative abundance of a certain type of modification to a certain type of process. Next, X chromosome inactivation also includes some trimethylation at H3K9 or H3K27. So, yeah. So these are the different types of processes which it can influence. Certain mechanisms underlie the effect of histone post-translation modification and the recruitment of histone modifying enzymes. So, for example, histone post-translational modifications or PTMs can act either directly, either they can affect the nucleosome interactions, like you can see on the left. If you zoom in here, you'll be able to see that certain regions, when they are modified, are affecting the internucleosomal interaction as well, which I previously mentioned in this schematic, where you can see uh, the relative distance or the gap between two subsequent nucleosomes is very minimal, which forms a tighter chromatin fiber. Whereas in contrast, for the second case, you can see that the internucleosomal interaction is also very, very low, therefore making it a good target for transcriptional activation and opening up DNA for global transcriptional activation as well. So that was one example. Apart from that, an indirect method is also via promotion or prevention of the binding of some reader proteins. For example, you can see that if there is a certain modification which is allowing or which is preventing the recruitment of some histone readers or the readers of some epigenetic marks, then that can allow or prevent a certain region from getting um, expressed or can actually repress that region. Like you can see here, um, I think this is the recruitment. Yeah, the pink reader is getting recruited at this locus and is recruiting it at some neighboring loci, whereas the presence of a certain modification is preventing the reader from getting recruited at that locus. Both possibilities actually exist. Next, histone modifications, a deeper look taken forward, involves the histone lysine crotonylation, which I've already mentioned is an evolutionarily sort of conserved post-translational epigenetic modification, which involves the reversible covalent attachment of a crotonyl group. Now, what is this crotonyl group that we keep talking about? It is also a subtype or a type of an acylation or an acyl group addition. Commonly, what we understand by acylation is the addition of acetyl group from acetyl coenzyme A as the donor, like we've already discussed. But if instead of that, from a crotonyl coenzyme A donor, if a crotonyl group gets added onto lysine, then that will lead to crotonyl lysine formation, like you can see here. The type of enzymes which can actually do it are also different. We'll come to it, it's mentioned here. Histone lysine protonylation, an evolutionarily conserved post-translational epigenetic modification, involves the reversible covalent attachment of this crotonyl group to the epsilon amino group of the lysine side chain. And histone lysine decrotonylation involves the removal of that same group. Some histone acetyl transferases also have this histone crotonyl transferase activity, like you can see here. Hats like P300, CBT, which is a hat, GCN5, ESA1, we saw these. And some decrotonylases also are actually functional as histone deacetylases like HDAT1, 2, 3, HDAT8, SERT1, SERT2, SERT3, etc. Earlier studies showed no correlation between the overall levels. Okay, we'll come to that actually later. This brings methylation. We'll finish this part before that. When we are talking about crotonylation, then one important thing to understand is where do we actually get this substituent group for the side chain to be actually added. Like for example, we understand from where acetyl coenzyme A is added. Similar to that, from the glycolytic pathway or downstream of the glycolytic pathway, there's a choice that has to be made between the directions in which it can further shift. So, for example, give me a moment. Yeah. So, for example, the TCA cycle here is generating citrate. From the citrate, you get acyl coenzyme A, which can work. And from there, two possibilities exist. Either a crotonate group will get added, generating crotonyl coenzyme A, or an acetate group will get added, generating acetyl coenzyme A. So the enzyme also that works here is similar or the same, which is ACSS2. And downstream of that, you'll be able to see which predominates. Let us read this first. The cellular concentration of acetyl coenzyme A and protonyl coenzyme A determines the degree of histone protonylation and acetylation. The metabolic production of acetyl coenzyme A and protonyl coenzyme A is shown in the above pathway. You have glucose as the initial starter molecule, which gets oxidized to acetyl coenzyme A in the mitochondria with the help of its ATP citrate lyase or the ACL enzyme. This is the ACL enzyme. So from citrate, which is the byproduct of the TCA cycle, ACL converts it into something which can get modified further. Acetate and crotonate, both of them belong to short chain fatty acid family, which are converted to their cognate acyl coenzymes catalyzed by ACSS2, the acetyl coenzyme A, or the acyl coenzyme A synthase 2 or synthetase 2. Crotonyl coenzyme A is the byproduct of fatty acid and amino acid metabolism. So these are generated physiologically within the cell. The crotonyl coenzyme A population is generated as a byproduct of fatty acid metabolism and amino acid metabolism. Whether crotonyl coenzyme A is generated from crotonate or other pathways is something which is still unknown. This is a possible hypothesis, but mostly you find this to be a byproduct of other pathways. The higher the ratio of crotonyl coenzyme A to acetyl coenzyme A, the easier it is actually the stoichiometry which predominates here. Depending on the stoichiometric distribution of both of these types of substituent groups, one of them will have a higher propensity to bind to the group, and it to bind to the acetyltransferase or acyltransferase, and accordingly that modification will be more preferably or more commonly added onto the DNA, and therefore that will predominate. So it depends upon the availability. Right. The histone acetyltransferases, or the HATs, like P300 CBP, like we have already mentioned, and MOF, they also have, just give me a minute. They also have HCT activity, which is histone crotonyl transferase activity. Class 1 HDACs and certain members of the sirtuins family, which are SIRT1, SIRT2, and SIRT3, they also play an important role in the HDCR activity. Besides, or the histone decrotonylase activity. Besides, the chromodomain protein, which is CDYL, inhibits histone crotonylation by converting crotonyl coenzyme A to beta hydroxybutyrate coenzyme A. Eats to AF9 MOZ, or MORF, DPF2, and KCR, and DPF2 are KCR readers, which is lysine crotonylation or crotonylated lysine readers that regulate various functions like DNA damage, repair, spermatogenesis, stem cell biology, and the like. Up, Apart from this, yeah, so both of these are usually activating marks, but apart from that, early studies showed or suggested that there is no correlation between the overall level of histone methylation and transcriptional activity. However, the effect of histone methylation is more complex than that of acetylation. Why so? Partially, that is because of the existence of these marks in three distinct states on both arginine and lysine residues, like RME1, which is monomethylation, dimethylation, and trimethylation, and lysine residues. The effect of histone methylation are site-specific and unlikely to directly affect nucleosomal structure. Okay, so this is one major difference. I don't have a side-by-side comparison of both these types of modifications, but majorly speaking, first of all, one of them is activating, the other is inactivating, or it is repressive in nature. The second thing is that one of them can exist in symmetrical, asymmetrical states, which is the methylation mark, and it can exist in these states on both lysine and arginine. The effect of histone methylation is also site-specific. It doesn't alter nucleosomal structure, whereas acetylation can actually do so. Um, yeah, so majorly these are the key differences. Now, one of these majorly characterized histone post-translational modifications that has gene transcription is the histone 3 or H3 K4 trimethylation. H3K4 trimethylation. What do we mean by this context? This post-translational modification is enriched in the promoter of most eukaryotic genes. Just a second, there is somebody calling me. Yeah. So this post-translational modification is enriched in the promoter of most active genes in eukaryotes, peaking around the transcription start site, where both its peak strength and its breadth correlate with transcription. What do we mean by this? For this, you need to have some concept of how these peaks are actually generated. So, yeah, while we'll discuss this later on, but basically what you understand from ChIP-seq or ChIP in general is the fact that regions which are bound by certain proteins or genomic loci which are bound by certain proteins can actually be isolated and can be studied. How so? Again, this relies on the concept that any protein bound region would be less accessible or inaccessible to a restriction enzyme, and therefore that region can be digested. Now, any region which is not digested can then be isolated. And in previous class, what we had seen was that those regions or those DNA sequences can be electrophoresed and can be studied. But in contrast, one very useful technique that also has much more sophisticated modifications involves the sequencing of those genomic sequences or those DNA sequences, making it a very useful technique in the long run. So when those regions are sequenced, the more number of sequences that support the presence of that region, the higher the peak. That means more accurate or more support enhances a higher and a sharper or more pointed peak. And the wider it is, the more that entire region has gotten protected because it is a stretch of nucleosomes that have actually bound that particular site. Now, then in today's time, you can actually get antibodies specific for one type of methylation of one type of lysine of one particular histone. So antibodies have become extremely specific, which has made it very convenient and very practically doable to actually carry out ChIP-seq of such precise modifications. And therefore, if you look at H3K4ME3, here you can see this is the difference between them. You have H3K4, normal lysine 4 with one methylation, with dimethylation, or with trimethylation. And if you perform chip sequencing, you can see blue, green, and yellow tracks of different types of chip-seq which has been carried out. And you can see that more the read density or the highest read density can actually be seen for H3K4, the highest peak. And the location of this is at two loci. One is right upstream of the active gene, and one is at the enhancer sequence. And yeah, so that actually shows that these are regions where activation has taken place, or at least where H3K4 ME3 modification is present. Um, yeah. So H3K4ME3 actually enables the recruitment of transcriptional machinery, and therefore it has a very positive or it acts as a very positive addition to transcriptional activation. It actually can facilitate transcriptional activation. It's not directly probably not going to do so, but it is helping, aiding, or allowing it. Secondly, H3K4ME3 dependent recruitment of TAF4, which is transcription initiation factor TF2D subunit 4, is also considered to promote the expression of some P53 target genes. However, um, some evidence from functional experiments in a lot of model systems have actually suggested that it is not a necessary a requirement for most transcription to actually proceed, but instead the local writing of H3K4MA3 can activate expression, modestly activate gene expression in a very context dependent manner. Therefore, the precise role till date remains unclear, but what it is likely to do is that it reinforces transcription, as transcription dependent recruitment of H3K4ME3 methylating complexes has been demonstrated in several systems. Also, H3K4ME3 influences transcriptional consistency, which means that it is inverse because it is inversely correlated with noise in gene expression levels. Let us understand this a little bit slowly. The deposition of this mark reinforces transcription. More the mark is like, or the presence or the abundance of this mark, it aids or helps transcription, like we have previously mentioned. Why so? The transcription dependent recruitment of these complexes has been observed in multiple model systems. That is why. Another possibility that is considered is that H3K4 trimethylation is the abundance of H3K4 trimethylation is inversely correlated with stochastic variation in the gene expression levels. So it is inversely correlated with noise in gene expression in contrast to H3K41, Me1, and Me2. So trimethylation is actually influencing the efficiency of transcription, which means the rate or the fidelity with which gene expression can actually be carried out is helped by H3K4 trimethylation. So these are the different variations. Um, in mammals, H3K4 trimethylation can be maintained during transcriptionally quiescent states, such as in mature oocytes and sperms, and in fertilized embryos before zygotic genome activation. This phenomenon is actually consistent with a long-term function of this mark downstream of gene expression, such as a role in epigenetic memory. What do we mean by that? We mean that sometimes it is not that the presence of this mark is going to lead to immediate transcriptional activation, but instead it is actually helping the maintenance of a transcriptionally quiet state. It's not repressive, it is something which has paused transcription in that duration at that point of time. How does it help? It helps because this has previously been studied in Drosophila, where the trithorax H3K4 methyltransferase complex has actually been studied. And it showed that epigenetic memory or retention of epigenetic memory in embryos or in the gametes is actually necessary. This hypothesis also gained traction from reprogramming work that was performed in Xenopus, which is in frogs, that showed that H3K4 trimethylation is necessary for the memory of active transcriptional states. So the presence of H3K4ME3 at certain loci in a quiescent state, in a transcriptionally paused state, is necessary so that when the fusion of the oocyte and the sperm occurs and a fertilized zygote is actually formed, within that an immediate switching on of the transcription can be carried out. So it's sort of preparing the cell in a manner that it now knows that these are regions which need to be actively transcribed. So you're leaving an epigenetic memory or you're leaving an epigenetic mark. Again, this is another way of looking at the term mark, but it is effectively that is what it is doing. This hypothesis was also supported from Xenopus, from some genetic reprogramming or chromatin reprogramming work that was performed in Xenopus embryos, frog embryos, and in mammals, H3K4ME3 at retained nucleosomes within the sperm seems to be actually important for establishing gene expression patterns and developmental capacity in the resulting embryos. So it is actually retained in certain nucleosomes within the sperm tissue, and that actually helps in switching on or establishing gene expression patterns expression patterns in the eventually formed embryo. This memory function seems unlikely to be unique to H3K4ME or methylation, as other PTMs are also present. So, histone PTMs have a major role to play in epigenetic inheritance. Again, this is the field which is actively being researched in today's time. If it is something of interest to you, you should certainly look upon it. It is going to be an immense thing to study. It is not just necessary to study diseases, it's necessary to study development, because a lot of times development can give you answers to many diseases, particularly to cancer. And so, yeah, it's an active area of research and you should certainly look into it. This is an sort of a 3D representation of what histone modifications actually do look like. So you can see that certain residues carry certain modifications, like for example, H3K122, succinylation, H3K115 acetylation, tyrosine phosphorylation, etc. And you can see that if you look at the images we were looking at till now, they seem to be something which is the modifications that we talked about seem to be present within the N-terminal tails, which are projecting out. But what is also important is that apart from the tails, some interactions also occur within the core and the inter interhistone PTM interaction, which means the interactions between these modifications also have a role to play in regulating how that histone is going to be controlled, what are the changes that are going to occur, and how is it eventually going to affect gene expression and gene regulation in the long term. So again, this I just wanted to, touch upon this topic because these are very there's advanced topics with a lot of a lot of impact, and these are areas that you need to be a little more comfortable with before we can delve into it. We will go into it when we discuss these techniques in more detail, but for now I'm just going to just introduce you to some omic techniques which can be used to study the histone modifications and their localization, their genomic localization, where a particular histone modification is present within the genome can actually be studied. So first method, which I already briefly mentioned, is the use of ChIP-seq. We will, again, don't worry, we'll discuss all of these techniques later on. If you look at the course handout, it's an elaborate course. We are only 1/3 of the way along it. But yeah, we're going to discuss it in a lot of detail. So ChIP-seq, the principle of ChIP-seq is that you actually have to cross-link a certain region of the genome, which is bound by the protein, to the protein which has bounded. In this case, the histone which has bounded. And then you subject it to sonication or micrococcal digestion to get rid of all of the non-specific regions. Or you can go for a non-cross-linked option, which is N-chip, so that it doesn't bias anything, and then subject it to micrococcal digestion. Single nucleotide resolution is possible with ultra low input material, which is EULI N-chip. For many years, ChIP-seq was the gold standard. Countless reference data sets, many chip-grade antibodies are available. X-chip is well suited, which is the cross-linked chip, is well suited for transient interactions. So depending upon your objective, you can use this technique to your advantage. Some disadvantages of this technique are the possibility that it has high background noise. Some standard protocols that require a lot of cellular input, very high sequencing depth. X-chip cross-linking can mask epitopes recognized by antibodies, and it's a very time-intensive protocol. Um, ChIP-seq takes around a week to complete, 5 days on an average to complete. So, and it is a very expensive technique. It's by chip-seq, I mean only the chip part of it. The sequencing part is, of course, going to be very expensive, but just by itself, the amount of antibody, the amount of reagents that go into this technique, they are immense and it takes a lot of time. But yeah, it's a wonderful technique to get a picture of the, you know, the chip or the DNA protein interaction of your interest within the cell. A global picture can be obtained at one go. Second, you can also do chip PCR, which is what is more commonly done when your objective is not to get a global picture, but instead to look at your own region of interest. Second, you can go for cut and run, which is cleavage under targets and release using nuclease, where recombinant MNAs fuse to protein A and or protein G binding to specific antibodies against a post-translational modification of your interest. It avoids, I'll come into details about more of this. So basically you use some regions which are fragmented, so which are cut. So it involves the use of a transposase. It involves the use of an MNase, which is a recombinant version of the micrococcal nuclease, which is fused to protein A or G, which is able to bind to your antibody of interest. And that antibody is specific to your post-translational modification of interest. It avoids or it bypasses DNA cross-linking, DNA fragmentation. It reduces background noise. It's possible with very low input. It's a much faster technique. Only low sequencing depth is required, and is very useful when you're doing some single cell analysis. Cut and run chip is often used to assess histone post-translation modification co-occupancy. MNA's digestion needs very careful optimization. That is a challenging thing. In addition to cleaving DNA, it can also, that is far away but close in 3 dimensions, because MNAs can do that. Because MNAs, the way in which we previously discussed, the way in which it binds DNA is, like it sort of sits on the DNA, straddles the DNA, and then cuts it like bolt cutters. So if DNA is close in 3 dimensions, that DNA can also sometimes get chopped. So that needs to be optimized. And transient interactions can often be missed because cut and run optimization is not always available for this particular method. I think this is our last slide. Yeah. Then you also have cut and tag. This has a transposase. The previous one I talked about was cut and run. Again, don't worry, we'll discuss these techniques in more detail. This is just an intro class. Recombinant TN5 transposase, which is fused to protein A, protein G, binding to specific antibodies. No cross-linking, no library preparation, a sensitive method, relatively easy workflow. It can be performed either at single cell level or can be used for multiple chromatin targets. PTMs in the same assay, single cell, genome-wide, spatial cut and tag is also possible. TN5 enzyme biases TN5. It preferentially tags accessible chromatin. Potential background is possible from mitochondrial DNA. So these are some disadvantages. Chromatin integration labeling followed by sequencing, which is chill-seq. So it combines immunostaining, transposase tagging, and linear amplification for low input PTM profiling. It is applicable to very low cell numbers owing to RNA-mediated amplification combined with cell biology assays. Some bias of the enzyme is possible. Sometimes fixation is required. Relatively a very long protocol, poor mapping efficiency necessitates high number of sequencing reads. And finally, a very different technique. I would not like to go into much detail about it. We'll discuss it in the next class. Direct methylation with long read sequencing, which is dimello-seq, which directs methylated adenosine or capped DNA to antibody protein A fusion coupled to long read sequencing. We have introduced all of these topics. That is why I proceeded in a certain manner so that you are not completely shocked when we come to this part. It has the ability to map very highly repetitive regions, concurrent protein and or PTM mapping with DNA cytosine methylation, multiple binding events on single molecules can be determined. very low sensitivity, and is not very useful for low cell numbers. So these are just some examples of some of the techniques which can be used. With this, we will end today's class. Please keep in mind that we have discussed um, some topics that have only been touched upon in today's class. We have discussed the introduction to epigenetics, the role of histone modifications, the significance of histone variants and how they can physiologically affect it, how, in fact, even histone modifications have a role to play in affecting disease, in affecting regulation. Let me just switch it on. Yeah. Um, how it has a role in yeah, physiologically regulating certain processes inside the cell. Sometimes histone modifications also coordinate with DNA methylation, like we have previously discussed. So these are all of the different possibilities that we have discussed till now. What I would suggest is that to not be very biased by certain topics, because these are not very textbook coded. These are mostly it's an amalgamation of what you will find as a traditional textbook material, and also linking it to information that is going to make it more relevant for you if you are interested in academia, if you're interested in research, if you are a researcher, or if you're pursuing PhD. So you want to be up to date with what is there in the world at the moment, and also integrate it with techniques that you will traditionally find in books. Also, keep in mind that every couple of days you'll find some new paper which has standardized some new technique. Don't always be intimidated by that, because people are constantly trying to improve A, the cost, B, the efficiency, and B, the amount of information which we can get out of a single technique, because that is going to make it a lot more cost effective. So that is an approach that every researcher actually has. So read everything, but don't be intimidated by it. Don't think that it's something you are not, very handsy with. If it is something you would like to discuss, please let me know. We'll certainly take it up. But overall, our idea is to A, understand epigenetics, B, understand what role does it have to play, why are we so bothered about it. And also, please keep in mind that here I'm only discussing epigenetics in context of gene regulation, in context of disease causation. But the field is immense. Its applications, like you saw in that combinatorial circular graphical abstract, that it has very important roles to play in a large number of processes, which we are not even touching upon, because then that would become a very specific course where we only talk about epigenetics, and that's not our goal. Our goal is to look at the gene, genome, and the epigenome. And specifically because our focus previously has also been on high throughput sequencing techniques, that's also something that I want to, keep you in touch with, because we'll come back to it eventually also. So yeah, that is the idea, that you use the cutting edge technology which is available today. You understand the practical applications of these techniques, you understand the shortcomings of these techniques, and then use it for your objectives, your hypothesis, and your experimental designing in the future. So with this idea, I would like to end today's class. I am hopeful of a better mm, of some attendance in the next class, and of some better discussions in the future. Thank you. Have a good night.